Good morning, students. Myself, Dr. Ravasada, Assistant Professor in Department of Microbiology, Government City College, is here with a topic on phase contrast microscope and fluorescence microscopy. Let's begin with phase contrast microscope. Phase contrast microscope was first described in 1934 by Dutch physicist Fritz Jernik. It's a contrast enhancing optical technique that can be utilized to produce high contrast images of transparent specimens, such as living cells, usually in culture, microorganisms, thin tissue slices, fibers, glass fragments, and subcellular particles, including nuclei and other organelles. Using this microscope, unstained living cells can be observed. These unstained living cells absorb practically no light. Poor light absorption results in extremely small differences in the intensity distribution in the image. This makes the cells barely or not at all visible in a bright field microscope. Phase contrast microscopy is an optical microscopy technique that converts phase shift in the light passing through a transparent specimen to brightness changes in the image. When light passes through cells, small phase shifts occur which are invisible to the human eye. In a phase contrast microscope, these phase shifts are converted into changes in amplitude, which can be observed as differences in image contrast. Phase contrast microscopy is another type of light microscope. It uses a special adapter that slows down the wavelength of light by 1/4, resulting in a phase shift. The phase shift results in the cell having a different refractive index than its surroundings. The light passes through the organisms versus the background differently. Organisms have a halo blue background. This is a diagram showing phase contrast microscope. Light, the different parts of phase contrast microscope are light, annular diaphragm, condenser, specimen, objective, and faceplate. We can see two important parts of phase contrast microscopes, that is annular diaphragm and faceplate, which help in producing the desired phase contrast image. Annular diaphragm and faceplate. With the help of the annular diaphragm and the faceplate, the phase contrast is obtained in this microscope. This is obtained by separating the direct rays from the diffracted rays. The direct light rays passes through the annular groove, whereas the diffracted light rays passes through the region outside the groove. Depending upon the different refractive indices of different cell components, the object to be studied shows a different degree of contrast in this microscope. Advantages of phase contrast microscope: small unstained specimens such as living cells can be seen, as staining leads to killing the microorganism. The staining process itself kills the microorganisms. So this particular this microscopy is used in observing living cells without staining. It makes highly transparent objects more visible. Examining intracellular components of living cells at relatively high resolution. Example, the dynamic motility of the mitochondria, mitotic chromosomes, and vacuoles can be observed using phase contrast microscope. It made it possible for biologists to study living cells and how they proliferate through cell division. These are some of the images of phase contrast microscope in comparison with bright field microscope. On one side you can see human chick cells. The image produced with bright field microscope, the first one. Below one is phase contrast microscope of HeLa cells. Then on the other side, you can see a comparison between a bright field and the phase contrast microscope of two different cells. Here are some of the images produced by phase contrast microscope. Diagram C is shown Pseudomonas, D is D-sulfomaculum, and below you can see Rhodosperlum rubrum. Limitations of phase contrast microscope. Phase contrast condensers and objective lenses add considerable cost to a microscope, and so phase contrast is often not used in teaching labs except perhaps in classes in the health professionals. To use phase contrast, the light path must be aligned. Generally, more light is needed for phase contrast than for corresponding bright field viewing, since the technique is based on the diminishment of the brightness of most objects. Next, moving on to the second type of microscope, fluorescence microscope. A fluorescence microscope is an optical microscope that uses fluorescence and phosphorescence instead of or in addition to reflection and absorption to study properties of organic or inorganic substances. Principle of fluorescence microscopy. Fluorescence is the emission of light by a substance that has absorbed light or other electromagnetic radiation, while phosphorescence is a specific type of photoluminescence related to fluorescence. Unlike fluorescence, a phosphorescent material does not immediately re-emit the radiation it absorbed. While the fluorescence microscope was devised in the early part of 20th century by August Kohler, Carl Richet, and Heinrich Lehmann, among others. In continuation of principle of fluorescence microscopy, most cellular components are colorless and cannot be clearly distinguished under a microscope. The basic premises of fluorescence microscopy is to stain the component with dyes. Fluorescence dyes are known as fluorophores or fluorochromes. These are the molecules that absorb excited light at a given wavelength and after a short delay emit light at a longer wavelength. The delay between absorption and emission is negligible, generally in the order of nanoseconds. Emission light can then be filtered from the excitation light to reveal the location of the fluorophores. This is a diagram which is showing a real-time fluorescence microscope, and the other side you can see a diagram showing the mechanism how this microscope works. The main parts of this microscope are mercury arc lamp, which emits a light, and the light emitted by the mercury is received by the excitation filter, which helps in passing only the blue light. The blue light is sent to the sample. The sample receives the blue light and it sends back green light through dichroic mirror. This dichroic mirror allows only green light to pass through it and prevents any remnant blue light around it. And this green light is received by the emission filter directed to the detector. So these are some of the pictures produced by fluorescence microscopy using fluorescent dyes. Like in the picture you can see there are different colors. The organs of these cells, internal organs of these cells are dyed with different fluorescent colors and which can be easily observed under a microscope. Advantages of fluorescent microscopy. It is the most popular method for studying the dynamic behavior exhibited in live cell images. This stems from its ability to isolate individual proteins with a high degree of specificity amidst non-fluorescing material. The sensitivity is high enough to detect as few as 50 molecules per cubic micrometer. Different molecules can now be stained with different colors, allowing multiple types of molecules to be tracked simultaneously. Limitations. Fluorophores lose their ability to fluoresce as they are eliminated in a process called photobleaching. Photobleaching occurs as the fluorescent molecules accumulate chemical damage from the electrons excited during fluorescence. Cells are susceptible to phototoxicity, particularly with short wavelength light. Furthermore, fluorescent molecules have a tendency to generate reactive chemical species when under illumination, which enhances the phototoxic effect. Unlike transmitted and reflected light microscopic techniques, fluorescence microscopy only allows observation of specific structures which have been labeled for fluorescence. Thank you.